Yeah, good morning. Today we are going to start with two chapters. We're going to start with respiration and complete two chapters. The first chapter for 15 minutes will be discussing about the summary of the chapter called respiration. In the last 15 minutes, we'll be discussing a little bit about hormones. Without wasting a single time, we'll start with respiration. All of you know that respiration is a process which could be divided into aerobic and anaerobic. In aerobic respiration, there is a complete breakdown of glucose into CO2 and water, therefore more energy is liberated. Whereas in anaerobic respiration, there is a partial breakdown of glucose either into ethyl alcohol or lactic acid, therefore the amount of energy released is very less. So we'll start with aerobic respiration. That is a very important point. We'll start with the first phase of aerobic respiration, which is glycolysis, where we convert glucose into first glucose 6-phosphate. And the enzyme name is kinase, and I utilize 1 ATP. Next, glucose 6-phosphate is converted to fructose 6-phosphate. Type of reaction isomerization. Enzyme name is isomerase. Fructose 6-phosphate to fructose 1,6-diphosphate. I utilize 1 more ATP. Type of reaction phosphorylation. Enzyme name kinase. This 6-carbon compound is going to break into DHAP and PGAL. This type of reaction is aldolase. Aldolase catalyzed. Type of reaction cleavage, because 6-carbon compound is breaking into 2,3-carbon compounds. DHAP cannot undergo further oxidation. D for do not undergo further oxidation. Easy way to remember. That is converted to PGL. So one PGL is formed from here, other from here. So the number of PGL is 2. This type of reaction is isomerization, because this is DHAP, this is ketone, this is aldehydes. Ketones and aldehydes are functional isomers. Well, you know pretty well about that. Now from here onwards, seriousness increases with regard to the importance with regard to your NEET exam. 2 molecules of PGL, remember this, is first converted to 2 molecules of 1,3-diphosphoglyceric acid. What did you study in chemistry? Aldehydes are converted to acids. That can happen on oxidation. Yes, oxidation means loss of hydrogens. Those are accepted by NAD to form NADH2. How many molecules of NADH2 form? 2. How many hydrogen atoms are lost? 2 2s are 4. 4 hydrogen atoms lost from 2 PGL. What is hydrogen atom otherwise called? Redox equivalent. How many redox equivalents are lost from 2 PGL? 4. Because 2 2s are 4 hydrogen atoms. 1 hydrogen atom is 1 redox equivalent. 4. But if a question is asked, how many redox equivalents are lost from each PGL? The answer is 2. 1,3-dipga is converted to 2 molecules of 3 PGA. Phosphoric acid is there the first and third carbon, here only on third carbon. Means there is a loss of phosphate accepted by ADP to form ATP. What type of reaction it is? Dephosphorylation. But in this process, ATP synthesized, therefore it's also called substrate level phosphorylation. Very important point, children. In your NEET, such type of questions are asked. This is the first energy yielding step, substrate level phosphorylation reaction. All right, next, what is the meaning of substrate level? Substrate is donating a phosphate to produce ATP, honoring the substrate for its outstanding generosity. You call it as substrate level phosphorylation. 2 molecules of 3 PGA to 2 molecules of 2 PGA. This type of reaction is intramolecular shift, positional isomers. Enzyme name not isomerase, mutase. 2 molecules of 2 PGA is converted to 2 molecules of PEP, in which 2 molecules of water is lost. Dehydration. Enzyme name enolase. PEP is converted to 2 molecules of pyruvic acid. 2 ATP are formed. Type of reaction dephosphorylation or substrate level phosphorylation. Very easy one to remember. Now the question you're going to ask me is, how many ATP are formed by substrate level phosphorylation? 2 plus 2, 4. Last year NEET question. How many ATP utilized? 2. If they ask you gross ATP formed, 4. Net ATP, 4 minus 2, 2. How many substrate level phosphorylation steps? Steps, 2. How many substrate level phosphorylation reactions? 2 here, 2 here, 4. Reactions are 4, steps are 2. All right, number of ATP formed 4. Number of ATP utilized 2. Net ATP 2. How many NADH2 formed? 2. Remember this. Amden, Meyerhoff and Paranas discovered these reactions. This is common for both aerobic and anaerobic. In anaerobic, this pyruvic acid, utilizing this 2-NADH2, is converted to lactic acid. That occurs in lactobacillus and also in your muscle cells when you run, fatigue. In yeast, these two molecules of pyruvic acid first loses 2 molecules of CO2 to form 2 molecules of acetaldehyde, and 2 molecules of acetaldehyde utilize this 2-NADH2 in order to form 2 molecules of ethyl alcohol. That is called alcoholic fermentation. There also there is no net NADH2 because utilized. In both lactic acid fermentation and alcoholic fermentation, net NADH2 will be 0. 2 are produced in glycolysis, utilized there. Net CO2 in lactic acid fermentation 0 because pyruvic acid is directly converted to lactic acid. Whereas in alcoholic fermentation, net CO2 formed is 2 because pyruvic acid is first converted to acetaldehyde, then to what is called ethyl alcohol. That point is a very important point to remember. See that you don't make any mistake with regard to this. That's a very, very important point. Next, we'll go with aerobic respiration. Chalo. You may ask me why you gave that much importance this to NADH2. What is the significance of this 2-NADH2? I'm going to talk about the electron transport system directly straight away, utilizing this 2-NADH2 to produce ATP. H2, it has got 2 electrons and 2 protons. My job is, imagine I'm NADH2. I'll go to mitochondria. Then I'm going to throw my electrons down because I have got 2 electrons. If I take one molecule here, I have got H2, 2 electrons. I lose my 2 electrons. When my 2 electrons go downhill, somebody should be there to take these electrons. Oxygen says, I will take these electrons. You throw. I am NADH2. I am throwing my electrons. Who is accepting? Oxygen. Oxygen have a greater ability to accept electrons and also protons. It is an ultimate hydrogen acceptor. And who is the ultimate electron and proton donor or hydrogen donor? I am NADH2. When I throw my electrons and protons, when the electrons go downhill to oxygen, passing through complexes, energy is liberated. Naturally, when electrons go downhill, energy is liberated. That energy I am going to use to first pump the protons, pump from lower to higher. Naturally, energy is needed to pump. Once I accumulate enough number of protons after pumping, I'll carry them through ATPase, ATP synthase to generate ATP. This process is called oxidative phosphorylation. Who is the man of the match here? I am man of the match because I am NADH2. Ultimately losing my electrons downhill. When the electrons go downhill, energy is liberated. Protons are pumped through complex 1, 4 protons. To complex 3, 4 protons. Through complex 4, 2 protons. 10 protons are pumped into perimitochondrial space. From there, the protons go again back to matrix through ATP synthase. Whenever 2 protons move from higher to lower, energy is liberated to synthesize 1 ATP. Out of 10 protons, 6 protons will move. 3 ATP are formed for each NADH2. I am going to explain that very, very fast here. Please listen what I am saying here. Did you understand? Look here. I am drawing the mitochondria first. This is perimitochondrial space. Concentrate here. This is cristae. And this is matrix. A, perimitochondria. B, cristae. C, matrix. So we'll take this. This is perimitochondrial space. This is cristae. And this is matrix. Okay. Chalo. Now what I do? On the cristae, this is complex 1, which contain FMN as a prosthetic group. And also contains 6 iron sulfur proteins. This is complex 2, which contains cytochrome B and cytochrome C1. Which contain 1 iron sulfur protein. This is complex 3, children. I'm sorry. This is 1, 3. Then this is complex 4, which contains cytochrome A1 and cytochrome A3. Complex 4. This is again very important. And this, all of you recognize, this is complex 5. This is called F0 and this is called F1 complex. And you know that there are 2 mobile electron carriers. It is a lipid ubiquinone embedded in the cristae. Cytochrome C, it's a mobile protein present towards perimitochondrial space. Chalo, how many NADH2 we produce in glycolysis? 2. I want to start with 1 NADH2. 1 NADH2 I take. 1 NADH2. I take it to matrix. Listen what I'm doing. I am losing 2 electrons to complex 1. From here the 2 electron goes to ubiquinone. Yes. For every 2 movement of electrons downhill, energy is liberated. That will pump 4 protons to perimitochondrial space. Pump 4 protons to perimitochondrial space. Easy way to remember. The downhill movement of electron release energy. That will pump 4 protons through complex 1 into the perimitochondrial space. What is the other name for complex 1? NADH dehydrogenase. Why? Because it is causing dehydrogenation of NADH2, oxidation of NADH2, and reduction of a mobile lipid ubiquinone. What happens next? 2 electrons. Ubiquinone accepts 2 protons to form a compound called ubiquinol. Naturally. What happens next? Ubiquinol now loses 2 electrons to cytochrome C and loses 2 protons actually, but your textbook says 4 protons to perimitochondrial space. So ubiquinone, I repeat again, ubiquinone accepting 2 electrons and 2 protons. Your textbook says 4 protons to form ubiquinol. Ubiquinol loses 2 electrons and loses 4 protons to perimitochondrial space. From B, the 2 electrons goes to C1, C1 to C. Let me explain about complex 3. What is the name of complex 3? Cytochrome C reductase. Because by giving 2 electrons to cytochrome C, it is causing the reduction of cytochrome C. Next, how many protons pass through complex 3? 4, just like 1, uphill. How many electrons pass through complex 4? 2, like this. That's it. And how many iron sulfur proteins? 1. Cytochrome C is a mobile lipid towards perimitochondrial space, don't forget that. From C, the 2 electron goes to A1 to A3, and ultimately to half molecule of oxygen. Again, is the electron going downhill? From here to here it is downhill. When the electrons go downhill, here also energy is liberated. And that energy will pump how many protons? 2. Through complex 1, how many protons pumped? 4 for each NADH2. Through complex 3, how many protons pumped? 4. Through complex 4, how many protons pumped? 2. Total, how many protons are there in perimitochondrial space? 10. Is it a reservoir of protons now? Yes. Now what? Out of 10, look here, 2 protons, they move, generating 1 ATP. Another 2 protons move, generating 1 ATP. Another 2 protons move, generating 1 ATP. So total, only 6 protons move out of 10 to generate what is called how many ATP? 3 ATP. It is very clear that 1 NADH2 is getting oxidized and is responsible to synthesize how many ATP? 3 ATP. Like that, how many NADH2 are produced in glycolysis? 2. Therefore, the number of ATP formed by oxidative phosphorylation will be 6. So, glycolysis, substrate level phosphorylation, gross 4. Oxidative phosphorylation, 2 NADH2, 6. Some of you want to know how 6. 1 NADH2 is going to produce 1, 2, 3 ATP. 2 NADH2 produce 6 ATP. What is it called? Oxidative phosphorylation. Where it occurs on mitochondrial inner membrane called cristae, which is acting as a proton reservoir, perimitochondrial space. How many complexes participate? 1, 3, 4 and 5. How many complexes participate in the movement of both electrons and protons? 1, 3 and 4. How many protons move through complex 1? Uphill, 4. Complex 3 uphill, 4. Complex 4 uphill, 2. Total, how many protons for each NADH2? 10. How many move back? 6. How many ATP formed? 3. Because 2 proton movement down the gradient, by breaking this gradient, release energy. For here, energy is needed. For here, energy is liberated. For 2 proton movement, enough energy is liberated to synthesize 1 ATP, 6 protons, 3 ATP. That completes electron transport system, which is looking very fine. What I'll do with 2 molecules of pyruvic acid. 2 molecules of pyruvic acid. Now I convert to 2 molecules of acetyl coenzyme A, which is a 2-carbon compound. Children, there is a loss of hydrogen, 2 NADH2, there is 2 CO2, magnesium is needed for this reaction. This reaction is also called link reaction. Enzyme name pyruvic dehydrogenase. This is the connecting link between glycolysis and Krebs cycle, 2-carbon compound. This is aerobic respiration. 3-carbon pyruvic acid is converted to acetyl-CoA. Now let us write how many ATP by substrate level phosphorylation, 0. Oxidative phosphorylation, 6. Why? All of you know this. This is complex 1. This is complex 3, complex 4, complex 5. From complex 1, 4 protons. Complex 3, 4 protons. Complex 4, 2 protons. 10 protons. 6 will move back. The number of ATP will be 3. 1 NADH2, 3 ATP. 2 NADH2, 6. That's it. That's about link reaction, which is looking very fine. Now it is time for us to talk about the Krebs cycle. 2 molecules of acetyl coenzyme A is going to combine with 2 molecules of oxaloacetic acid. So easy way, I told you CIO. It first forms citric acid, I. It forms isocitric acid, O. It forms oxalosuccinic acid, then A, S and S. Then it forms alpha ketoglutaric acid, S. It forms succinyl coenzyme A, S. It forms succinic acid, then FM, O. All right, succinic acid to fumaric, fumaric acid to malic, malic to oxaloacetic. C, citric, isocitric, oxalosuccinic, alpha ketoglutaric, succinyl coenzyme A, succinic, fumaric, malic and oxaloacetic. What type of reaction is link reaction? Oxidative decarboxylation. Remember that. This is 2 carbon combining with 4 carbon to form 6 carbon. This is synthase, joining. This is 6 carbon isomerization. Easy way to remember. From here onwards, the troubles start. Therefore, I want you to go back to link reaction. Type of reaction is oxidative decarboxylation. Break it into 2. Oxidation, 2 NADH2. Decarboxylation, 2 CO2. Oxidative decarboxylation. So easy to remember. Oxidation first, decarboxylation, followed by oxidative decarboxylation. Now, succinyl-CoA to succinic acid. This coenzyme A is a high energy compound. When it is lost, energy is liberated. That energy helps in coupling GDP and organic phosphate to form GTP. GTP further break, releasing energy, which help in combining ADP and inorganic phosphate to form ATP. So ultimately, everything is 2 molecules, children. Ultimately, how many ATP formed? 2. Ultimately, 2 ATP formed by substrate level phosphorylation. Then succinic acid to fumaric acid. Here, 2 FADH2 formed. And fumaric acid to malic, 2 water utilized. Here also, oxidation, 2 NADH2. Your textbook says how many NADH2 formed in Krebs cycle for 1 molecule of glucose. Easy way shall I tell you, draw 4 lines like this and draw a line here. 2 NADH2, 2 NADH2, 2 NADH2, 2 FADH2, and this is the 2 ATP formed by substrate level phosphorylation. See, if they ask you in Krebs cycle for 1 molecule of glucose, how many NADH2 formed? 2, 2, 2, 6. If they ask you for Krebs cycle means you should take 1 turn. So 1, 1, 3 NADH2. So how many NADH2 formed in Krebs cycle? 3. How many NADH2 formed in Krebs cycle for 1 molecule of glucose? 6. How many FADH2 formed in Krebs cycle? 1. How many FADH2 formed in Krebs cycle for 1 molecule of glucose? 2. How many ATP formed in Krebs cycle by substrate level phosphorylation? 1. How many ATP are formed in Krebs cycle for 1 molecule of glucose? 2. Now I want to write how many ATP by substrate level phosphorylation in Krebs cycle for 1 molecule of glucose? 2. Oxidative phosphorylation in Krebs cycle for 1 molecule of glucose, 2, 2, 2, 6 NADH2, 6 3s are 18, 18 ATP formed. Why 18? You already explained. Now don't forget, there are 2 FADH2. what we can do with this 2-FADH2, now let me explain this. All right, same. This is perimitochondrial space. This is cristae and this is matrix. Complex 1 not involved. Complex 1 is not involved for FADH2. This is complex 2. This is succinic dehydrogenase. This is the Krebs cycle enzyme attached to cristae. It oxidizes succinic acid to convert it into fumaric acid. Succinic acid loses hydrogens accepted by FAD, which is a prosthetic group in complex 2, accept those hydrogens to form FADH2. Okay? Now what does FADH2 do? It is going to lose 2 electrons to ubiquinone. All of you know that. For every 2 electrons, ubiquinone accept 2 protons to form ubiquinol. Come on children, you know all this. This is complex 3. Ubiquinol loses B to C1, 2 electrons, and in this process, textbook says 4 protons to perimitochondrial space. C1 to C. Then you know that this is A1, A3, ultimately to half molecule of oxygen. How many protons here? 2 protons. So 4 protons to complex 3 and 2 protons to complex 4, 6 protons, out of which only 4 protons will move. Therefore, how many ATP synthesized? Only 2. It's very clear that 1 FADH2 produces 2 ATP by oxidative phosphorylation. 2 FADH2 produce 4 ATP, as simple as it. Which complex is not participating? Complex 1. How many protons get transported to perimitochondrial space for each FADH2? 6. How many protons move back? 4. How many ATP synthesized? 2. The number of ATP synthesized from each FADH2, 2. From 2 FADH2, 4. Let me wind up this chapter. Glycolysis, substrate level phosphorylation, gross 4, oxidative 6, total 10. Link reaction, substrate level phosphorylation 0, oxidative phosphorylation 6, total 6. Why 6? Because there are 2 NADH2. Why 6? 2 NADH2. Each NADH2 3 ATP. Krebs cycle for 1 molecule of glucose. Substrate level phosphorylation 2, oxidative phosphorylation 6 NADH2. So 18, 2 FADH2, 4. So it comes to 24. 10 plus 6, 16. 16 plus 24, the answer is 40. This is the total ATP formed. Out of 40, 2 are utilized in the first and third reaction. Therefore, the net ATP will be 38. This is turning out to be an easiest chapter. So what we did, children, in glycolysis, link reaction and Krebs cycle, we produced NADH2s and FADH2s. Then what did we do in electron transport system? The high energy protons and electrons, means hydrogen atoms of NADH2 is lost to oxygen. When the hydrogen atoms are lost to oxygen, all right, then naturally the electrons go downhill, releasing energy. And that energy will pump the protons against the concentration gradient. From complex 1, 4, complex 3, 4, complex 4, 2, total 10 protons if it is NADH2. If it is FADH2, complex 1, 0, complex 2, 0, complex 3, 4, complex 4, 2, out of 6, 4 will move back through complex 5, producing 2 ATP. In this way, we have produced what is called the ATP, what is needed by oxidative phosphorylation. Children, respiration is seen in all organisms because it's a source of energy released. Respiration is seen both in green and non-green cells. Respiration is an ultimate source of energy released. All right, because whatever the foodstuffs we eat, that is broken down to energy. When the CC bonds are broken, energy gets released. And that energy is converted to an energy currency called ATP. And that is stored in our body. Whenever we need energy, this ATP breaks, releasing that energy. And respiratory intermediates can also help in the synthesis of other macromolecules. If glycerol wants to enter respiratory pathway, it enters at PGAL. If fatty acid wants to enter the respiratory pathway, it enters at acetyl-CoA. If amino acids wants to enter the respiratory pathway, it can enter at pyruvic acid, enter at acetyl-CoA, or enter at any intermediate if Krebs cycle. Also understand the entry points are also the exit points. It means that respiration intermediates, the carbon skeletons of respiration, can also help in the synthesis of glycerol, fatty acids, and amino acids. Therefore, respiration is rightly called amphibolic pathway. CO2 by O2 is said to be RQ value. For carbohydrates, RQ value is 1. 6 CO2 by 6 O2. Fats and proteins, all of you know, they are poorer in oxygen. When they are poorer in oxygen, C51, H98O6 is tripalmitin. For 51 carbon atom, there are only 6 oxygen atoms. Because they are poorer in oxygen, they require more oxygen for oxidation. So less CO2 by more O2. RQ value will be less than 1.7. For amino acids also, they are poorer in oxygen, they require more oxygen for oxidation. RQ value is 0.9. And for organic acids, they have rich in oxygen. Because these compounds are rich in oxygen, they require less oxygen for oxidation. Therefore, the RQ value will be more than 1. You and I have respiratory organs, but not plants. Reason why plants don't have respiratory organs. 1. Plants do not present greater demand for gaseous exchange because their metabolism is far less when compared to animals. 2. The distance oxygen need to travel in plants is very less because every plant part can take care of their own oxygen needs. 3. The cells inside, even though they are deep-seated, they have large intercellular spaces which can act as storehouse of oxygen, exposing at least a part of the cell to oxygen present in the intercellular spaces. Therefore, there is no big problem as far as taking oxygen is concerned. Don't say that stomata and lenticels are respiratory organs. They are not respiratory organs. They help in respiration. Respiratory organs are gills, moist skin, and lungs, which are exclusively seen in animals, not in plants. The amount of energy released per 1 gram of any particular respiratory substrate is not same. Fats are going to release approximately 9 kilocalories, unlike carbohydrates and proteins, which is approximately 4 kilocalories. Even for taking oxygen, I require energy. So energy is needed even for breathing. Is breathing seen in all organisms, starting from Monera to Animalia? Yes, every organism has to breathe. Then only they can take oxygen. Then only they can completely oxidize the foodstuffs and release energy. That is the entire story of a chapter called Respiration. Today's agenda is also to discuss one more chapter called Hormones, but unfortunately I cannot complete, but definitely I'll manufacture some time to bring hormones in tomorrow's lecture apart from our regular class. What we require hormones is 10 minutes, not more than that. It is more memory based, and I'll tell you the easy way to remember. Don't forget to join again tomorrow at 10 o'clock to discuss about the next important chapter. రేపొద్దున ఇగో మీ క్లాస్లో ఈ మాస్టర్ ఈ పాఠం ఎగ్జామ్ చెప్పాను అన్నట్టు ఉండాలి. మైక్ ఆన్ ఉందా? తప్పే ఉన్నాడు.
అది నేను ఒకటే చెప్తాను.
మీకు ఎంత అయితే అంత చెప్పండి. ఇంక్రీజ్ ద పిచ్. కొంచెం వాయిస్ ప్లీజ్. అండ్ స్పీడ్ ఆల్సో యూ కెన్ ఇంక్రీజ్.
Good morning. So today let us summarize the topic chemical control and coordination. Regarding the chemical control and coordination, it is mainly mediated by the chemical messengers known as hormones. Nervous system generally provide the point-to-point interaction, and it is short-lived, and of course it is a rapid transmission. But when compared to the nervous system, in endocrine system, for the production of hormones it takes time, and the response also will have more time, and it is long lived when it comes to the endocrine system. Next, let us talk about the hormones. Hormones are secreted by a specialized glands known as endocrine glands, which are said to be the ductless glands. Hormones are the chemical messengers. They are non-nutrient chemicals produced in trace amounts. Hormones are target specific in nature, because the targets are having receptors for the hormones. Hormones are highly effective. Though they are produced in less amounts, the physiological activities performed by the hormones are very effective. But once the action is over, the hormones will be degraded. When we take the example of liver, liver can degrade the steroid hormones after their action. That is the reason why the bile juice consists of breakdown products of steroid hormones. Majority of the hormones, they are produced at one place and act in another place. First, the hormones are released into the intercellular spaces. From there, they will get into the blood. Blood carry the hormones to the target tissues. But some hormones, they are produced and act at the same place. You can call them as local hormones. The best example for the local hormones, gastrin. So this is the introduction related to the hormones. Let's talk about endocrine glands of our body. Related to the brain, we have hypothalamus is endocrine, pituitary endocrine, pineal endocrine. In the neck, thyroid and parathyroid are endocrine. Above the heart, thymus gland. Above the kidneys, adrenal gland. In the loop of the duodenum, pancreas. In males, testis. In females, ovary. These are all organized endocrine glands. But when it comes to the heart, liver, gastrointestinal tract, they are said to be the non-organized endocrine glands. Let's begin with the hypothalamus. Hypothalamus is the part of diencephalon of a forebrain. This hypothalamus consists of specialized cells for the secretion of hormones known as neurosecretory cells. The hypothalamus is connected to the pituitary gland. It is connected to the adenohypophysis of the pituitary gland through portal circulation. It is connected to the neurohypophysis of the pituitary gland through hypophyseal tract. The hormones that are produced by the hypothalamus that are going to the neurohypophysis and stored there, the examples for those hormones are oxytocin and vasopressin. Vasopressin is also known as anti-diuretic hormone. But other hormones of hypothalamus, when we take that are going to the adenohypophysis, they are said to be the releasing hormones and inhibitory hormones. One example I will tell you for that, growth hormone releasing hormone, which is also known as somatocrinin. When growth hormone releasing hormone, if it reaches the adenohypophysis, then adenohypophysis prepares the growth hormone, which is called somatotropin. Growth hormone inhibitory hormone, you can call it as somatostatin. When adenohypophysis receives the somatostatin, then adenohypophysis stops the production of growth hormone. Like that, there are many releasing and inhibitory hormones can be produced by the hypothalamus and will go to the adenohypophysis, like thyrotropin releasing hormone, because TSH is thyrotropin. Melanotropin releasing hormone, because MSH is melanotropin. Corticotropin releasing hormone, it stimulates the production of corticotropin called ACTH. Gonadotropin releasing hormone, it stimulates the production of gonadotropins like FSH and LH. So that means hypothalamus is regulating the pituitary gland, but majority of the hormones of pituitary gland are tropic hormones. They regulate the other endocrine glands. As pituitary is regulating the other endocrine glands, you can call it as master gland. But hypothalamus is regulating pituitary so that it is master control center. So let's begin with the next gland, that is pituitary. Pituitary gland is attached to the hypothalamus with the help of a stalk known as infundibulum. This pituitary is accommodated in a cavity formed by the bone of the cranium. That cavity name is the sella turcica. Initially, the pituitary gland consists of three lobes like pars distalis, pars media and pars nervosa. But later pars media is fused with the distalis. Now you can pars distalis and media together called adenohypophysis, that is anterior pituitary. Pars nervosa is posterior pituitary, nothing but neurohypophysis. Adenohypophysis is connected to hypothalamus, neuro is also connected to hypothalamus. But neuro is connected to the hypothalamus through the nervous tract. Hence, the neuro is said to be under the direct neural regulation. Let us see the hormones of adenohypophysis. First, pars distalis. The pars distalis produces, the first hormone is growth hormone. Functions of growth hormone, it is required for the cell division and protein synthesis. Deficiency of growth hormone in children may lead to a disorder known as pituitary dwarfism. And pituitary dwarfism, physically they are dwarf, but mentally and in other activities they are normal. But excess secretion of growth hormone in children may lead to gigantism, where there are tall limb bones can be seen. In adults and middle-aged people, excess secretion of growth hormone leads to a disorder known as acromegaly. It is hard to diagnose actually, but the symptoms of this are facial disfigurement. If it is not treated properly, it may lead to the premature death. The next hormone is prolactin. Prolactin is required for the secretion of milk and growth of the mammary glands. The next is TSH, thyroid stimulating hormone, which is also known as thyrotropin. The thyroid stimulating hormone produced by the adenohypophysis, especially from the pars distalis, it will go to the thyroid gland and stimulates the thyroid gland for the secretion of iodothyronins. It is not for thyrocalcitonin, only for iodothyronins. Next, ACTH, adrenocorticotropic hormone, also known as corticotropin. It stimulates the adrenal cortex mainly for cortisol, Next, FSH and LH. When we talk about FSH, follicular stimulating hormone, it mainly acts in the testis for spermatogenesis. And FSH in ovaries function is follicular growth, ovarian follicular growth. But when it comes to the LH, when it reaches the testis, it acts on the Leydig cells of the testis, stimulates the Leydig cells for the production of androgens. These androgens, FSH, LH, in turn, they are involved in the regulation of spermatogenesis. Next, LH functions in females. The first function of the LH is in the middle of the typical menstrual cycle, LH surge is required for the rupturing of Graafian follicle. You can call it as ovulation. And LH maintains the corpus luteum. So, these are the main functions of LH in females. Next, MSH, melanocyte stimulating hormone secreted by the pars intermedia. It stimulates the melanocytes for the secretion of melanin and increases the pigmentation. These are the hormones of adenohypophysis, both distalis and pars media. When we talk about the hormones of pars nervosa, it is not secreting any hormones. It can store the hormones that are produced by the hypothalamus like oxytocin and vasopressin. When we talk about the functions of oxytocin, levels mainly required for the contractions of smooth muscles of uterus at the time of delivery, and also required for the contractions of smooth muscles present in the mammary glands for the milk ejection. Let us talk about ADH, antidiuretic hormone. Antidiuretic hormone function is whenever the body fluid volume is low, ADH is released into the blood, it will go to the distal convoluted tubule. In the distal convoluted tubule, the ADH involved in the reabsorption of water followed by salts from the filtrate, thereby increases the blood volume, thereby increases the GFR. And one more function of this ADH is, it is also known as vasopressin, because it will constrict the blood vessels of the kidney, thereby increases the blood pressure. So, by increasing the blood pressure also it can regulate the kidney functions. Deficiency of ADH may lead to excess loss of water through the urine, that is known as diuresis. Diuresis is the main character of a disorder known as diabetes mellitus. In diabetes mellitus, there is excess urination without glucose. Diabetes insipidus. In diabetes insipidus, there is excess urination without glucose. So, these are the hormones of pituitary gland. All the hormones of hypothalamus are peptide in nature, hormones of pituitary are also chemically peptide in nature. The next gland is pineal gland. Pineal gland is attached to the epithalamus of diencephalon. Pituitary is attached to hypothalamus of diencephalon, pineal is attached to the epithalamus of diencephalon. Pineal gland secretes a hormone called melatonin. Melatonin is an amino acid derivative hormone. When we talk about the functions of melatonin, you remember M cube. So, 1 M for it regulates the circadian rhythms. So, before going for that M cube, the main function of the melatonin is it regulates the circadian rhythms, sleep wake cycles of an individual. 24-hour day cycle will be regulated by this. Along with that M cube, 1 M for it regulates the metabolism, second M for it regulates, it has influence over the menstrual cycle, third M it is related to the melanin functions, especially it is said to be it decreases the pigmentation of an individual. So, along with this circadian rhythms and MQ functions, the two more functions of melatonin are it regulates the body temperature and also involved in defense reactions. The next gland is the thyroid gland. So, when we talk about the thyroid gland is present between the voice box and trachea, and it has two lobes connected with each other by a bridge called isthmus. Ventrally it has isthmus, dorsally you can see the two lobes only. Each lobe consists of many lobules known as the follicles. The follicles of thyroid gland are lined by the follicular cells. Between the follicles there is a connective tissue known as stroma. Stroma consists of parafollicular cells. These are follicular cells, these are parafollicular cells. Follicular cells secretes T3 and T4, triiodothyronine and tetraiodothyronine. Parafollicular cells secretes a hormone called thyrocalcitonin. T3, T4 are iodothyronine, thyrocalcitonin is a protein hormone. In this, T4 is called thyroxine. Let's talk about the functions of thyroxine. Thyroxine regulates the basal metabolic rate of an individual. It is required for the metabolism of proteins, carbohydrates and lipids. Thyroxine influences the uptake of oxygen by the cells. Thyroxine involved in the generation of energy in the cell, hence it is called calorigenic hormone. And thyroxine is osmoregulatory in nature because it influences the sodium potassium pump. It is also required for the production of RBC because it influences the bone marrow. So, these are the main functions of thyroxine, and the thyroxine also regulates the body temperature. Whenever the thyroxine levels are low due to the deficiency of iodine, that leads to a disorder called goiter. Enlargement of the neck is the main symptom. But less secretion of thyroxine in pregnant females may lead to a condition called cretinism. Less secretion of thyroxine in non-pregnant females may lead to some menstrual issues, and less secretion of thyroxine in adult men and women may lead to a disease called myxedema, in which you can see a weight gain and also low metabolism in the body. But excess secretion of thyroxine leads to a disorder called exophthalmic goiter, also known as Graves' disease. The two reasons for the excess secretion is, one is thyroid cancer, second one is the autoimmune nature. Due to the excess secretion of thyroxine, the metabolic rate increases and there will be weight loss in those persons. Next, let's talk about parafollicular cells. Parafollicular cells secretes a hormone called TCT, thyrocalcitonin, which is a protein hormone. Thyrocalcitonin main function is it decreases the blood calcium levels by moving the calcium from blood to the bone. When calcium get into the bone and deposits there, you can call it as mineralization. So, TCT is involved in mineralization. So, after the thyroid gland, next one is parathyroid gland. On the dorsal side of the thyroid glands, thyroid lobes especially, on each lobe 2 parathyroid glands, 4 parathyroid glands can be seen. The parathyroid glands produces a peptide hormone called parathormone. Parathormone function is to increase the blood calcium levels by 3 ways, I can tell you, main 3 ways. One, it can move the calcium from intestine to the blood, dietary calcium, with the help of vitamin D. It can move the calcium from the filtrate, kidney filtrate to the blood, that is called reabsorption. It can also move the calcium from bone to the blood, that is called resorption. By absorption, by resorption, by reabsorption, calcium is pulled into the blood, thereby the blood calcium levels are increased. So, this is about parathormone, which is a peptide hormone. Whenever the parathormone is low, that may reduce the blood calcium levels. So, low blood calcium levels may lead to a condition known as tetany. So, after the parathyroid, the next one is thymus gland. So, thymus gland location is, it is located above the heart, in front of the aorta, just behind the sternum, between the 2 lungs. It is a lobular gland. The size of the thymus gland varies depending on the age. It produces hormone called thymosin. Thymosin is a peptide hormone. The functions of thymosin, it is mainly involved in the maturation of T lymphocytes. But T lymphocytes, there are 2 types of T lymphocytes majorly here, TC and TH. TC lymphocytes are involved in cell mediated immunity directly. TH lymphocytes can influence the cell mediated. TC lymphocytes, by activating the B lymphocytes, they can influence the humoral immunity also. But humoral immunity is mediated by the antibodies. So that we can say T lymphocytes will not produce antibodies, but T lymphocytes can promote the production of antibodies. So, in old age, as there is a shrinking of thymus gland, there will be less secretion of thymosin that may lead to some immunity issues. Next, so after the thymus, the next gland is adrenal gland. A pair of adrenal glands are located on the kidneys. Adrenal glands are having 2 divisions. Outer portion is called adrenal cortex, inner is called adrenal medulla. Adrenal cortex and adrenal medulla. See, if it is the kidney, this is the adrenal gland. So, this is cortex, this is medulla, cortex and medulla. The cortex outer to inner is having 3 zones, G, F, R, zona glomerulosa, zona fasciculata, zona reticularis. and all these three collectively produces the hormones called corticosteroids. And adrenal medulla produces amino acid derivative hormones called epinephrine and norepinephrine, also known as adrenaline and noradrenaline. When we talk about the glomerulosa, glomerulosa produces some mineralocorticoids. The principal mineralocorticoid is aldosterone. Regarding the function of aldosterone, the first step is whenever the glomerular filtration rate is low or glomerular blood flow is low, the JG cells of the kidney produces an enzyme called renin. That renin activates angiotensinogen of the blood into angiotensin 1. The angiotensin 1 is converted into angiotensin 2 with the help of angiotensin converting enzyme. This angiotensin 2, it is a powerful vasoconstrictor. It will activate the zona glomerulosa of adrenal cortex, leads to the secretion of aldosterone. This aldosterone will go to the distal convoluted tubule, causes the reabsorption of salts followed by water. Thereby it increases the GFR, increases the blood pressure, increases the blood flow to the kidney. So, this is the function of aldosterone. Next, zona fasciculata produces a hormone called cortisol. Cortisol is the principal glucocorticoid. Functions of cortisol are very important, 2 or 3 times they asked in NEET. So, regarding the functions of cortisol, it is the glucocorticoid. It is involved in increasing the blood glucose levels. It increases the blood glucose levels by increasing the glycogenolysis, breakdown of glycogen, gluconeogenesis, synthesis of glucose from non-carbohydrate sources. That it also increases the proteolysis and lipolysis. And other functions of the cortisol includes, it is anti-inflammatory in nature. It is a immunosuppressor hormone, and cortisol is required for the cardiovascular and renal functions. And cortisol can reduce the stress. Along with that, cortisol is also included in the production of RBC. And last function of the cortisol is, cortisol inhibits the uptake of amino acids by the cells. These are the functions of cortisol. Deficiency of this cortisol leads to a disorder called Addison's disease. In Addison's disease, the glucose metabolism is disturbed, that may lead to the weakness and fatigue. Next one is reticularis. Reticularis produces cortical sex hormones. Cortical sex hormones are involved in the pubic hair growth, axial hair growth and facial hair growth. So, this is about adrenal cortex. When it comes to the adrenal medulla, medulla produces epinephrine and norepinephrine. These are not antagonistic, functions of both are same, also known as adrenaline and noradrenaline. These are the amino acid derivative hormones. And functions of epinephrine, norepinephrine are similar to the functions of sympathetic nervous system, that means emergency situation. When we talk about the epinephrine, norepinephrine, these are called fight or flight hormones, also called emergency hormones. They increases the heart rate, they increases the breathing rate, they increases the alertness, profuse sweating, there is piloerection because of release of these hormones. But to increase all these, they needs more glucose. For that, these hormones are also involved in the breakdown of glycogen and also converting the non-carbohydrates into glucose. That means glycogenolysis increased by these hormones, gluconeogenesis is also increased by these hormones. This is about epinephrine and norepinephrine. So, after adrenal gland, next one is pancreas. So, pancreas is located in the loop of duodenum. This is the pancreas. It will open generally into the duodenum. In the pancreas, there are 2 types of cells, pancreatic acini and pancreatic islets of Langerhans. Pancreatic acini are involved in the secretion of pancreatic juice that consists of enzymes. Islets of Langerhans are involved in the secretion of hormones. The 2 main varieties of islets of Langerhans are alpha and beta cells. Let us talk about beta cells. Beta cells secretes a peptide hormone known as insulin. When we talk about the functions of insulin, insulin is a hypoglycemic hormone. It reduces the blood glucose levels. How it reduces the blood glucose levels? By moving the glucose from blood to the body cells. The main target cells of insulin are hepatocytes and adipocytes. When glucose is moved into the hepatocyte, it is converted into glycogen and stored there. That is called glycogenesis. Insulin increases glycogenesis. And when glucose enters into the adipocytes, there it will be stored in the form of fat. So, that is lipogenesis. So, insulin increases glycogenesis and lipogenesis, but it decreases glycogenolysis, gluconeogenesis, proteolysis and lipolysis. Deficiency of insulin leads to a disorder known as diabetes mellitus, in which there is elevated blood glucose called hyperglycemia, there is appearance of glucose in the urine called glucosuria, and appearance of ketone bodies in the urine known as ketonuria. These are the main symptoms of diabetes mellitus. Next, after the insulin, next one is glucagon. The functions of glucagon are opposite to the insulin. Whenever the blood glucose levels are low, glucagon will be released, it increases the blood glucose levels, hence it is called hyperglycemic hormone. How it increases the blood glucose? By breaking the glycogen into glucose, that is glycogenolysis. So, glucagon increases glycogenolysis, of course it also increases gluconeogenesis, proteolysis and lipolysis, but it decreases glycogenesis. So, these are the functions that are related to the pancreatic hormones, and both insulin and glucagon are peptide hormones. Insulin is secreted by the beta cells, glucagon is secreted by the alpha cells. Next, let us talk about gonads. So, regarding the gonads, first testis. Mind the testis, the leading cells of the testis secretes androgens. The principal androgens are testosterone. The Sertoli cells of the testis secretes a hormone called inhibin. Let us talk about the functions of androgens. Androgens are involved in the regulation of secondary sexual characters of male and also required for the development of accessory ducts of male reproductive system. It regulates spermatogenesis. It is involved in the regulation of characteristics like facial hair growth in males, body hair growth in males. It regulates the male sexual behavior, aggressiveness of the male. These are said to be the anabolic steroids involved in the anabolism of proteins and carbohydrates. So, these are the main functions. Of course, these are also required for the skeletal growth of the male. So, these are the functions of androgens. When it comes to the Sertoli, Sertoli produces inhibin. Inhibin function is it inhibits the release of FSH, thereby regulates spermatogenesis. Next, let us talk about ovary. Regarding the ovary, suppose if it is the ovary, these are the growing follicles of the ovary and this is the corpus luteum of the ovary. The growing follicles of the ovary secrete estrogens. Principal estrogen is estradiol. The corpus luteum secretes estrogen, progesterone mainly, along with that it also secretes inhibin. So, inhibin is secreted by females, especially the granulosa cells of corpus luteum. Functions is same, it inhibits the release of FSH. In males, inhibin is secreted by Sertoli. But when it comes to the estrogens, the functions of estrogens, it regulates the female secondary sexual characters. It is regulating the oogenesis, it is regulating the development of mammary glands in females, sexual behavior in females. Of course, it is also required for the skeletal growth in females. It regulates the menstrual cycle in females. So, these are the functions of estrogen. One more point is estrogen is required for the regeneration of endometrium in menstrual cycle, especially in follicular phase. When it comes to the progesterone, three functions of progesterone you remember. The first function of progesterone is it maintains the pregnancy by preventing the contractions of uterus. Second function of progesterone is it is required for the differentiation of alveoli of mammary glands during pregnancy. Third one, it is also required for the lactation. Mucosa of the stomach is required for the gastric motility, secretion of pepsinogen and HCl. Next hormone is enterogastrone. Enterogastrone is secreted by the mucosa of small intestine. Its actions are opposite to the gastrin. Hence, these are antagonistic to each other because enterogastrone prevents gastric motility, inhibits gastric motility. Third hormone is secretin. Secretin is also secreted by the mucosa of the small intestine. Secretin acts on pancreas for the bicarbonates and water. The final hormone is cholecystokinin, secreted by the mucosa of the small intestine. Cholecystokinin acts on gallbladder for bile juice, acts on pancreas for pancreas secretion of, stimulates the secretion of enzymes from the pancreas. So, these are the functions of gastrointestinal tract. Majority of these hormones are peptide, hormones of non-endocrine glands. Now, let us talk about the mechanism of hormonal action. But before going for that mechanism of hormonal action, let me tell you the hormones are having two types of receptors related to their target cells: surface receptors, intracellular receptors. Hormones are classified into two categories: water soluble and lipid soluble. Peptide protein hormones plus amino acid derivative hormones comes under water soluble. Thyroid, especially iodothyronins, and steroid hormones comes under lipid soluble. Water soluble hormones cannot cross the plasma membrane of the target cell, hence they will go and bind to the surface receptor. Fat soluble hormones can cross the membrane, so that they will go and bind to the intracellular receptors. Next, water soluble hormones like peptide, amino acid derivative hormones, they work through the secondary messenger system. Next, fat soluble hormones, so directly they go and bind to the receptor inside the cell. Let us talk about one example for water soluble hormone mechanism and one example for fat soluble hormone mechanism. FSH is the example for water soluble hormone. This FSH target cell is ovarian cell. When FSH go and binds to the surface receptor of the ovarian cell, that hormone receptor complex initiates response 1 inside the cell. That response 1 is nothing but activation of certain enzymes involved in the formation of secondary messengers. And next is the generation of secondary messengers like cyclic AMP, calcium ions and IP3. And after that, that will initiate the biochemical responses, that means several enzymatic reactions. Finally, biochemical responses stimulate the physiological response inside the cell. The physiological response related to the ovarian cell with respect to the FSH is follicular growth, growth of ovarian follicles. So, they need a secondary messenger system. When we talk about one fat soluble hormone mechanism, I am telling you estrogen. Estrogen, the target cell is, let us take the cell of endometrium of uterus, uterine cell. So, regarding the estrogen, estrogen can cross the plasma membrane, estrogen can also cross the nuclear membrane inside the uterine cell. Then it will go and bind to the intracellular receptor. After binding to that, the receptor hormone complex, it is motile here, it will go and bind to a particular gene and regulates the gene expression. So, thereby that gene undergo transcription and produces mRNA, mRNA undergo translation and produces the protein. Those proteins are involved in the physiological responses like tissue growth and differentiation, that is nothing but thickening of endometrium. So, these are, this is the mechanism related to the fat soluble hormones. And in this lesson you better remember the antagonistic pairs, gastrin enterogastron antagonistic, insulin glucagon antagonistic, thyrocalcitonin parathormone antagonistic, oxytocin progesterone antagonistic, but epinephrine, norepinephrine are not antagonistic. ADH and ANF are antagonistic. So, when you concentrate on these points and revise the chapter based on this knowledge, you will require only a little time to complete the chapter. So, thank you.